Hi, this is Steve St. John, Professor of Psychology at Rollins College. This is the fourth in a series of videos on sheep brain dissection. Today we'll be doing coronal sections, which is a type of cross-section. Okay, so coronal sections basically refer to cuts that are perpendicular to the long axis of the nervous system. And so sections made by taking the knife, pressing it straight down all the way to the table are referred to as coronal sections. And when you're looking at the face of a coronal section, you're basically looking at the front, the view of the brain from the front. Therefore, you can see a lot of structures that are bilaterally symmetrical that are going to be on both the right side and the left side of the brain. Now we're going to cheat a little bit today. I'm not going to cut into this brain. I'm going to use this as a reference to show you where the slices have been made that I've already prepared. That way we make sure we get sections that show all of the key structures in the brain that we need to. And as usual, I'm going to present my tour going from front of the brain to back of the brain. Now in an earlier video where we were looking at the dorsal surface of the sheep brain, I defined this area here as cerebrum, as forebrain. This area here as the beginning of hindbrain, and there's also structures of the midbrain which are sort of buried down in here beneath the, between the cerebrum and the cerebellum. So as we go back, we'll be looking at forebrain, midbrain, and hindbrain. And more specifically, we'll be looking at structures in the anterior forebrain, where the cut is made about right here. Structures in the posterior forebrain, where the cut is made about right here. Structures in the midbrain, which would be a section through here. Structures in the front part of the hindbrain, which would be straight through the sort of thick part of the cerebellum. Structures in the posterior part of the hindbrain, the cut plane of section being right here, and then the spinal cord. We'll look at the spinal cord in coronal section as well. So there's 6 total sections that I'm going to be showing you. Anterior forebrain, posterior forebrain, midbrain, anterior hindbrain, posterior hindbrain, and spinal cord. Okay. So the first section we have is anterior forebrain. Again, the plane of section would be just through the front part of parietal lobe. You can certainly make coronal sections more anterior than this. The most anterior sections will contain only cerebral cortex. As you move back, you'll see structures that are visible on this particular section for quite a long distance in the sheep brain. Okay? And in this section, the section's dominated, more than half of the section is dominated by cerebral cortex. So cerebral cortex consists of these sort of loops of gray matter interspersed by regions of white matter, and usually you can get pretty good contrast of those two areas. As you know, if we flip it further, you can see the gyri and sulci on the surface of dorsal surface of the brain, which is the upper part of the cortex. And so all this is cerebral cortex. Specifically, this is parietal cortex over here, actually running all the way along the top. And then this area of cortex here that's kind of buried down below the surface of the cortex is called the cingulate gyrus. Some people call it the cingulate lobe of the brain. The dividing line between the cerebral cortex and the subcortex is a very high contrast, very vivid strip of white matter that runs across the midline of the brain. And that structure is called the corpus callosum. In an earlier video, when we made sagittal dissections, we learned to identify the corpus callosum. Corpus callosum connects areas of the left hemisphere with areas of the right hemisphere. So everything sort of ventral to the corpus callosum pretty much is subcortical forebrain. Below the corpus callosum and forming kind of they almost look like parentheses back to back with one another are these openings, these spaces, and those are the lateral ventricles. Again, when we did sagittal dissections, we identified the lateral ventricles as the major ventricles of the anterior forebrain. They were visible just below the corpus callosum, so we see them in the same location in coronal sections as well. Lateral to the lateral ventricles on either side, if you can sort of envision an egg-shaped region of gray matter, these are the basal ganglia, which consists of the caudate nucleus, the putamen nucleus, and some other subnuclei of the basal ganglia. And so these you can find lateral to the lateral ventricles. On sections with higher contrast, you can often see white matter running through the basal ganglia. This structure is called the internal capsule, and more specifically, it's axons of neurons of motor cortex that are running through the forebrain and eventually through the rest of the brain down to spinal cord to contribute to control of movement. So on this anterior forebrain section, we have cerebral cortex, including the cingulate lobe, the corpus callosum, the lateral ventricles, the basal ganglia, and the internal capsule. And again, that plane of section is made right about here at the beginning of the parietal lobe. If we move back posterior, we get a section that looks something like this. Okay, and if you compare it to the previous section, you'll notice that the cortex has gotten a little bit thinner, not quite as thick. The corpus callosum has gotten a little bit thinner as well, or a little bit less defined. And there, we sort of lost the basal ganglia. It no longer exists. We've also lost the parenthesis shape of these lateral ventricles. The lateral ventricles still do exist, but as you move more posterior in the forebrain, the lateral ventricles appear more dorsal in the section. And so this space here and here are the lateral ventricles. If you see some brown in the lateral ventricles, which I do, those are a gland called the choroid plexus, which makes cerebrospinal fluid. Okay, so cortex, corpus callosum, lateral ventricles. Here's some choroid plexus here that's very visible, but that's in a separate ventricle that I'll talk about in a second. This plane of section seems to have almost what look like sunglasses or something right in the more dorsal part of the section underneath the lateral ventricles, and that's the hippocampus. Hippocampus is a pretty dramatic structure in the sheep brain, and in the human brain for that matter. Ventral to the hippocampus is the third ventricle. The third ventricle runs along the midline. It's usually largest right underneath the hippocampus and forms a line. Sometimes you'll see sort of disconnected regions of ventricle along the midline. That's all third ventricle, and this choroid plexus is in the third ventricle. Lateral to the third ventricle is the thalamus and hypothalamus. And sometimes you'll see the optic chiasm below it, depending on how far back you are. So we have cortex, corpus callosum, lateral ventricle, hippocampus, third ventricle, thalamus, and hypothalamus. The dividing line between the thalamus and the hypothalamus is difficult to see. Sometimes you can see some white matter that seems to define a border between the thalamus and the hypothalamus, but they're sort of just right up next to one another. Okay? It can be a little confusing discriminating the basal ganglia, which we find in this more anterior section, from the thalamus and the hypothalamus, which we find on this more posterior section. They look like egg-shaped structures. They're large structures of the subcortex. There's a couple ways to tell the difference. One is the basal ganglia appears lateral to the lateral ventricles. So here we have a lateral ventricle. Lateral to it, that gray matter is basal ganglia. Here, where the thalamus is, we're no longer lateral to the lateral ventricles. We're lateral to the third ventricle, and we're ventral to, we're underneath the lateral ventricle. We're also underneath the hippocampus. So both of those landmarks can tell you that the structure here is thalamus and hypothalamus, whereas the structure here is basal ganglia. As we go more posterior and now make a cut sort of a coronal section through the occipital lobes of the brain, down through the front part of the cerebellum, and down into the brainstem area, then we'll get midbrain. And so here's our third section as we move back. This is midbrain. And you can see now it looks very different. The cortex is sort of separated from the brainstem region, whereas before the subcortical forebrain was connected to the cortex. So now that we're back as far as midbrain, the brainstem region is far enough back that it's actually sort of, this is overlying cortex. It's not connected to this region down here. All of this is midbrain. The midbrain is defined by a couple of landmarks. These bumps on the very top surface of the midbrain, or dorsal surface of the midbrain, those are the colliculi, the superior and inferior colliculus. Those bumps are much more easily seen from a dorsal view of the brain, but you can see them in cross-section as still having that bump-like appearance. There's also a very large round ventricle of the midbrain, which is the cerebral aqueduct. So superior colliculus, inferior colliculus, cerebral aqueduct. The gray matter around the aqueduct is referred to as the periaqueductal gray, which just means the gray matter around the aqueduct. So, and then we have regions of the tegmentum or possibly pons on the bottom part of the section. It's difficult to identify from this angle. So occipital lobe, colliculi, cerebral aqueduct, periaqueductal gray. As we move further back, we're going to start getting into the hindbrain. Now we are so far back that we're no longer cutting into cerebral cortex. Instead, we're back far enough that we're cutting into the cerebellum. Okay. And the cerebellum lies on top of the brainstem. Probably this is pons if we're far enough forward. This bottom part of the brainstem is referred to as the pons, more anterior part of the brainstem. The space in between the cerebellum and the pons is another ventricle. It's called the fourth ventricle. So there's not much to see here other than cerebellum, fourth ventricle, and what is probably the pons, although it could be the beginning of the medulla. As you go further back, cerebellum gets smaller and the brainstem gets smaller. Again, you still have fourth ventricle in between the cerebellum and what is now the medulla, or medulla oblongata. We're back in the very back part of the cerebellum now. So I don't know if you can see this section of spinal cord here. It's not much to look at, obviously. It's a very small structure, but you maybe can see that sort of dark dot in this dorsal middle of the section, which is the central canal of the spinal cord. Okay. Spinal cord there. So, quick little review. Anterior forebrain is characterized by a lot of cortex and the basal ganglia internal capsule, the lateral ventricles forming that unique kind of back-to-back parentheses shape, and the corpus callosum is usually very bright white. As you move further back in the forebrain, the cortex gets a little bit smaller, subcortex gets a little bit larger, and now the dramatic structure is the hippocampus, which look like goggles in the top surface. The hippocampus separates the lateral ventricles from the third ventricle, which is both here and here. Kind of makes like a T shaped. The subcortex here is thalamus and hypothalamus. Further back now, we're cutting through the occipital lobes, which hang out over above the midbrain, the colliculi of the midbrain, the cerebral aqueduct of the midbrain, and the periaqueductal gray. Further back, we've lost cortex. We're completely in hindbrain. We're cutting through the cerebellum. Cerebellum lies on top of the pons, and the ventricle in between them is the fourth ventricle. Still further back, cutting through the back part of the cerebellum, fourth ventricle still visible, and the medulla is visible. And somewhere between this section and the spinal cord, the fourth ventricle turns into the central canal, which is the tiny little ventricle that runs through the back part of the medulla and then all the way down to the end of the spinal cord.